So finally, I'm going to briefly talk to you about, well, what happens if we include mutations? So in all of the previous work, we were excluding mutation, and there are good reasons for that, which are discussed in the supplementary material of our preprint. But one particularly interesting case is where the treatment itself induces mutations. So for example, chemotherapy, which is mutagenic, or if there's some kind of plastic response in the sensitive cells, then we have this term mu, which is dependent on our drug dose, here denoted U, which is a treatment-dependent mutation rate. And so what I'm going to introduce you to here is some work that was done by Timo Kuusmanen, who is a master's student at the University of Helsinki, and I was somewhat involved in his master's project, which looked at what happens when one introduces this drug-induced mutation rate. And so these are the assumptions that Timo made. The key one here is on the right, that we have a linear dose-dependent mutation rate that increases with the treatment. And what's interesting here is that we can get evolutionary rescue, which was nicely introduced by Stefan in his talk. And in the evolutionary rescue case, what we see is that the sensitive population goes down, but there's a possibility that the resistant population can escape. And this is particularly interesting for us in the case of finite population size and stochastic escape. So Timo's paper has some very nice optimal control results, which show that the results he obtains in stochastic simulations really generalize. But what I'm showing you here is just an example of some stochastic results, which back up his main result, which is that when there is this trade-off such that the treatment eliminates sensitive cells, but also induces more mutation from sensitive to resistant cells, then we can find that an intermediate dose is optimal in terms of the probability of cure. In other words, in terms of minimizing the probability of evolutionary rescue. So just in conclusion, what I've shown is that containment strategies generally improve on MTD, maximum tolerated dose, even if resistance has no cellular fitness costs. It's a very general result. But the predicted gains crucially depend on the nature and the intensity of competition between cells. And what Timo has shown is that when treatment promotes mutation, then lower doses can also maximize probability of cure. And so we find that there is definitely reason to continue experimental and clinical trials of this kind of approach in cancer therapy. So I'd just like to thank the people I worked with, Yannick, who did who led the work in our bioarchive paper, Timo, whose thesis is being turned into a paper that should be up soon, Daniel Fisher and his lab, who carried out the experimental work that I talked about at the beginning, and Mike Hochberg, who was my supervisor at Montpellier when I carried out that study. And thank you to the funders shown here. Thank you.
Thank you, Robert. So there's one question in the chat asked, and that's, do resistant cells suffer more from competition because GR was strictly decreasing in S, while GS was only non-increasing in R?
Yeah, so these are the assumptions we need in order to obtain this result. So basically, so I'll just go back to show people what I was talking about then. So the general assumptions we make. Yeah, so essentially GS could be completely independent of R. It could be the sensitive cells are not affected by resistant cells. This is essentially, in order for these results to hold, the resistant cells need to be at some kind of a slight disadvantage. And the fact that we have a strictly decreasing here and a non-increasing for the sensitive cells captures the fact that sensitive and resistant cells are different in some way. So yes, the resistant cells need to be at least as badly affected as the sensitive cells by this competition. Does that make sense? And I mean, essentially what this is also capturing is that the main ecological interaction is competition here and not some kind of beneficial interaction. If you start having beneficial interactions, then this approach might not work.
Okay, we have one more question, and there's another question by Eduardo. So, Eduardo, please go ahead.
Yeah, hi, Eduardo Sontag here. No, I just wanted to mention that, especially what you were talking about at the end, we had a couple of papers with Jana Geberts and Jim Green, I don't know if you're aware of this, comparing drug-induced mutations or epigenetic changes with sort of more random mutations. And we analyzed from an optimal control point of view. In the deterministic case, we analyzed from an optimal control view which kind of induction you have actually changes what the optimal strategy is. So I don't know, it's just something that you might want to look into the relations to that.
Yes, I will certainly check whether we already cite those papers.
Oh, okay, all right, sorry.
I mean, there's a lot of work in these papers that I haven't discussed. So yeah, if not, then I will definitely be.
I look forward to reading your paper.
Thank you.
Good talk, very nice.
Okay, Mario, you are muted.
Thank you, Felix. So we have time for one more question. So it is further up, but it may be interesting to the audience here. So the question is, could you provide more intuition on why no fitness cost is needed? I think should be explained a little more.
Essentially, because of these assumptions we make here. So the idea is that the best way, well, the only way we have of suppressing the growth of resistant cells is through competition with sensitive cells. And if we have this assumption that the growth rate of resistant cells is strictly decreasing in the population of sensitive cells, then our best strategy is to maintain as many sensitive cells as possible so that we maximally suppress the resistant cells. And so I think this is expressed better in the preprint. We have a section on just this mathematical intuition, but that's the basic idea. And therefore, in general, there is no need for resistant cells to have a generally lower growth rate in the absence of competition. The key thing is that we have this competition going on. But what we do find is that if there is this fitness cost, which has been shown in various experimental systems to hold, then the clinical benefits can be much higher than in the absence.
All right.
Yeah.
Thanks, Robert, again.
Thank you very much, Robert. We can move to our third talk, and it will be Mario Center from